In this interview, I'm talking to Professor Jeffery Leigh from Sussex University. Jeff, thanks for coming and talking to me.
My pleasure.
I want to start off by asking you simply, what kind of scientist are you?
Oh, I'm an inorganic chemist, and that means I deal with really basic materials. Organic compounds are too complicated for me. I'm just dealing with the basics.
So a mixture of experimental and theoretical?
Primarily experimental, yes.
Right. Okay.
Yeah.
I'm interested in what got people into science. Were you interested in science as a schoolboy?
Well, I got into chemistry specifically at school when I've had my first chemistry lesson.
Right.
And the class teacher took some ammonia and some hydrochloric acid, these colourless liquids, and made these beautiful crystals of ammonium chloride, and then he sublimed them.
Right.
So you had colourless liquids giving crystals, and crystals actually going through the gas phase and forming again. I thought that was fantastic. So I was a chemist after that.
You were hooked by that moment.
Yes.
And some other things, presumably. Now, I want to talk to you about your work with the Nitrogen Fixation Unit, or Nitrogen Fixation Department, which was set up at Sussex here. And as far as I'm aware, it was a very interdisciplinary and international department. Can you tell me a bit more about that briefly?
Well, we were set up, and perhaps uniquely at that time, to study the mechanism of biological nitrogen fixation.
Okay. Which fixation means what?
Well, fixation, that is an alchemical word. It means really removing from the gas phase and somehow trapping on a solid.
Oh, okay.
So it's nothing to do with drugs. And if you want, people always talk about nitrogen fixation, which is simply taking the gaseous nitrogen and making compounds out of it.
Okay. So we live in a world, in an atmosphere that's got oxygen, carbon dioxide, nitrogen, traces of argon and helium. Now, we all know that the oxygen and the carbon dioxide are essential for life, but there's this thing called nitrogen, which also is essential. So can you tell us a bit about that?
Well, it's, as you say, essential for life. Nitrogen is an essential part of proteins, it's an essential part of DNA, it's found in all sorts of vitamins and other biological molecules. Yep. And the question which sometimes then arises is, well, how does it get there if we've got all this nitrogen in the atmosphere, which is very inert? How do you convert, or do you convert, nitrogen from the gas phase to these compounds?
Yeah. So I know on the Apollo missions, when they first went to the moon, they actually the capsule had pure oxygen, because humans can breathe pure oxygen.
Yes, yes.
So, but because it's difficult to make sure that the nitrogen was diluted properly. But then there was a terrible fire, I think, on one of the early, and so they had to make sure they put the nitrogen in. But nitrogen isn't just diluting the oxygen, is it? It's a much more important part of the whole growing process than that.
Well, it is, actually. If you want to know all the background, may I sort of point you to this book, which is called The World's Greatest Fix, and it has nothing to do with drugs.
No.
But ever since agriculture started, people have been aware that you can't go on raising plants in the same patch of soil generation after generation after generation, because the fertility drops. And it was discovered by really just by experience that there were certain things, and night soil is a primary one, but also horse manure, animal manure. The Romans were particularly fond of dove manure. I don't know how they found that out, but
they had a lot of doves, presumably. But this has been known for many, many thousands of years.
Yes, this has been known for a long time, but since people really had no concept of what nitrogen was, let alone oxygen or anything else, this was purely experimental, purely a matter of experience.
Yeah.
The other system which was developed, and which is again talked about in here, is crop rotation. And that's because there are certain plants which seem to be able to also restore soil fertility.
Right. So if you just try and grow the same crops week again and again and again, the crops basically after a time don't really grow.
That's correct.
But there's something in the natural. If you go to an island, if you come back to the UK, go back 100,000 years, it would have been full of trees and bushes, happily growing for thousands and thousands of years. So there must be something else in the soil that's...
Well, there is fixed nitrogen
Yeah.
in the soil, but it's a limited amount.
Yes.
And
So when you say that, so lightning or something can react, break up the nitrogen, form nitrogen compounds, it rains down.
Indeed, yes.
And they can be there, but this is a small
Yes.
amount.
But there are also, apart from what we've called nitrogen fixing plants, which is not particularly accurate, as we'll explain, there are also microorganisms living in the soil,
Right.
free-living microorganisms, which can fix nitrogen.
Right. So they're taking the molecular nitrogen, which is fairly inert, and in some way they're doing chemistry on it to basically make a manure or a fertiliser.
Yes, absolutely so.
Right.
And as chemistry developed, then of course people began to ask questions about this. I think one of the original stimuluses was that metal carbonyls, and nickel tetracarbonyl is always a classical example, which has a nickel atom with 4 carbon monoxide molecules attached. This is well known for a long time, some rather unpleasant material, but that's beside the point. And that is, as chemists say, isoelectronic with N2.
Right.
Because you've got an oxygen which has 1 more electron than nitrogen, and carbon which has 1 electron less. But overall, CO, carbon monoxide, is the same number of electrons as N2.
Right.
So why can't you stick 4 nitrogen molecules onto a nickel?
Yep.
And that was a basic kind of question we were trying to answer.
So before we come on to the chemistry, at Sussex was set up this nitrogen fixation unit, presumably to bring all sorts of people together to study this basic question.
Yes, yes.
So that was, can you tell us a bit about the history of the unit?
Yes, indeed. Well, it was recognised that the nitrogen fixation question was, shall I say, multi-dimensional. That sounds very, really a bit sophisticated. You shouldn't say things like that. But there was the straight chemistry. We don't know how the chemistry works. And then there was the biology, because we knew that there were microorganisms living in the soil, free as I've said, but also in the roots of the legumes, which are particularly well recognised group of plants, peas and beans, etc., which were also apparently able to fix nitrogen. So when we fix nitrogen industrially, which was a great breakthrough, I may say,
Yes.
we use high temperatures, high pressures, catalysts. We really have to
A big industrial plant.
We really have to push the system hard.
Yeah.
We can make ammonia
Yes.
and use that as a fertiliser.
Yes.
But these microorganisms don't seem to be constructed in quite that way.
They don't need high temperatures, high pressures. No.
They're working in the soil
Yes.
and in the presence of oxygen, which is a poison for these systems.
Yes.
So this was a very broad question, and the Agricultural Research Council, which, as it was at the time, decided that they wished to find out how the biological system worked, but it had to be treated from various points of view. It couldn't be just done by one man or one small group.
And Sussex, being a new university, seemed to be very good at these multidisciplinary things. We could take a diverse range of interests and people and bring them together. And that's what happened here at Sussex in the 60s, was it?
Yes, the early 60s, yes.
Yeah, and you were part of that, the whole group?
We were part of that. We built our own building here. And Colin Eborn, who was the first professor of chemistry, was instrumental in bringing us here. They had tried to set up the unit in Queen Mary College in London, but for various reasons that didn't work. And we were better off here, I think.
Yeah, yeah. Have you any particular part of that research that you want to talk about?
Well, yes, I'd like to talk about this business of trapping N2 on metals.
Right.
And
so the nitrogen in the air is fairly unreactive.
it is unreactive.
You've got to try and make that reactive.
That's absolutely right. So one of the first things that we set out to do, because we knew the biologists had shown us that, or the biochemists perhaps to be more accurate, they'd shown us that these biological systems needed metal atoms somewhere.
Yep.
Molybdenum or vanadium were known to be important, and there was always iron, but then iron is a bit misleading because it's used in so many ways in biological systems.
Yep.
So we looked at the carbonyls, as I've mentioned. There are lots of those. Why aren't there many, many more compounds of N2 just like that? Well, we were a bit embarrassed because other people discovered the first N2 complexes. One was a ruthenium compound, another was a cobalt compound, but they were shown to exist.
Right.
And eventually we got in on the act, and we made N2 complexes of molybdenum, tungsten and iron and ruthenium and osmium and, well, most of the transition metals of the periodic table. So we did very well. But then there was a second question.
Right.
Which was simply, well, you can bind the N2 to a metal.
Mm-hmm.
The metal has to be supported by various other groups, but they're not important in this discussion. How do you convert the N2 to ammonia?
Right.
Because
So then you've got a useful product that you can use.
Yes. The N2 compound itself is just a sort of an academic interest.
Yep.
If it just sits and looks at you.
Yeah. Yep.
So that was our next problem, and we found the first reaction of what we called coordinated N2.
Yes.
By accident, of
Right.
course.
Yep.
Like all good good discoveries, I think. We were trying to do something quite different. We were trying to take a tungsten compound with two N2 molecules stuck top and bottom like that, and we wanted to do oxidative additions because the N2s weren't doing anything except flying off as far as we were concerned.
Yep.
So we did a reaction with acetyl chloride,
Right.
which should have put a chlorine underneath and an acetyl group on top in place of the two N2s. And what we discovered eventually, much to our surprise, was we could put the chlorine underneath, but on the top we had the metal joined to a nitrogen, and then an acetyl group. So we had converted the N2 to a different compound altogether.
Right.
And that was the beginning of it.
Wow. Yes.
And later we discovered we could put hydrogens on there as well.
Is there any chemistry? I know we've got a slide of the chemistry.
Yes,
Can we talk that through?
Let's just look at that. Because this is an example, and probably in many ways, for me, the most interesting of all my work, but that's a bit personal. This is an iron compound here.
Okay.
So iron joined to two chlorines, and it doesn't matter what they are. You can reduce it with borohydride, and you can make a polyhydrogen compound. And these polyhydride compounds were also quite novel and very interesting. But this one reacts with N2 to produce an N2 complex.
Right. So you can't do this in air, but do you just bubble the N2 through the solution? How do you actually do that?
Well, we work under N2 generally,
Okay.
so we
Yep.
just keep the
Yep.
oxygen
Yep.
away.
Yep. Okay.
Then if you treat that with acid, and that the alcohol is just a solvent,
Yes.
then the iron compound breaks down completely, and some N2 is generated, some H2 is generated, but the important thing is that some ammonia is generated.
Right. So in principle, you could take nitrogen from the air and through this reaction make an ammonia to make fertiliser for crops.
That's right, and rather nice to think of in a way. 20 years after we published this, some people have this year published another paper in which they've extended the chemistry of this to make the whole thing catalytic.
Oh, right. So you can just go round and round
So
and round
you can go round
and process
round.
more and more nitrogen.
Yes.
Fantastic. Thank you very much. Is there any principle or result or mathematical technique or something that you've discovered over the years as being particularly useful or helps you understand science? Or any observations?
Well, yes, this is really almost philosophy, but I've known many eminent chemists, including many actually Nobel Prize winners. And the thing that struck me about them all is the vast amount of information they have at their fingertips. They don't go and look it up.
Yeah.
They know it's there. And it means that if you see an observation or you make an observation, you can start to think, well, I know this happens there. Why doesn't it happen here? Yeah. And the second thing is that you've actually got to go and do it. You must work in the lab to test your ideas and your theories, because the way you make advances is not by confirming what you think is true,
no,
but by actually discovering what you think is wrong.
By doing experiments.
By doing experiments.
In the real world.
So I think it's important for every chemist to be involved in experimentation.
Jeffery Leigh, thank you very much.
My pleasure.